So let's go back to our early stages of development. Cell division takes place after fertilization, and at these early stages there's no growth. So every time the cells divide, they get smaller. So from 1 cell to 2 cells, they half in size. At 4 cells, they're a quarter of the size. And they go on dividing until they've reached the normal size for a human cell. As they do so, they form a ball, and this is called the morula. And then that ball will become hollow. And this stage is called the blastocyst. And the blastocyst is not a simple hollow ball like a tennis ball. There's a little cluster of cells on its inside, and that little cluster is called the inner cell mass. So the inner cell mass is marked in the diagram. The outer cells are trophoblast cells. And this is not just a difference in naming. Something extremely profound has happened here, because the baby that will be born will come from just a few of the inner cell mass cells, and all of the other cells that are present, including especially the trophoblast cells, will contribute to the extra embryonic membranes, such as the placenta. So this first division into 2 different kinds of cells is actually really important, and therefore understanding how this takes place might be a valuable clue to understanding the process of differentiation of one cell from another. So looking at these stages again, 2 cells to 4 cells, 8 cells, and then increasing numbers all the way up to the hollow ball of cells, the blastocyst, with the inner cell mass perched at one pole within the hollow mass of the blastocyst. And an extremely clever experiment by Professor Chris Graham explained how this might happen. What he did was to take some labelled cells from an embryo, it was a mouse embryo in fact, and he took a morula and placed the labelled cells in different positions within the morula. And what he found was that the cells that he placed in the centre of the mass would subsequently contribute to the inner cell mass. In other words, they'd be on the lineage that contributed to the baby. Cells that he placed at the edge of the ball of cells would contribute to the trophoblast. In other words, they would contribute to the placenta. And what this means is that cells had responded to their position by changing what they would turn into. So our question of what informs the DNA, in this case, turns out to be a positional signal reflecting where a cell lies with respect to all the other cells round about it. So that key distinction, that important decision about inner cell mass versus trophoblast, whether it was going to be part of the baby or part of the placenta, came from a positional signal. And that's a valuable clue to understanding the processes that underlie all differentiations.